Welcome once again to your biology class here at Meritnation. Hello Angie, hello Mera, hello Rebecca. How are you all? Hello Kashif, Pranav, Chinmay. Good evening to everyone. How are you, people? Abby Calp. Oh. I got 2 new people here. Fine, good, great, superb. Who's sad? Who is sad? Okay, you are spraying. Okay, do pay for me also. Hi Chinmay, looking happy. Prajwal, why you are sad? Okay, no, don't do that. Pray for us also. So, all of you are here. Could not solve my full maths paper and lost 5 marks. Oh, no problem. Because it's not the end of the day. If you know that answer, that means you are there. You are getting the full marks. You understand, Prajwal? It's not that doing in examination that is that is important. But what is important is that you know that question, you know that answer. That is much more important. You did it there, it's fine. If you didn't did it there, that doesn't take anything from you. It's only the marks, nothing else. You understand what I'm trying to say? It's all in here, not in the paper. You did practicals to examine cheek cells. Oh, great. You lost 2 marks, Humera. Why so? People are actually very upset of losing 2-3 marks. Okay, you did it long back. Okay. So Angie, how was your practical? Oh, it's not that you don't have to use such a force in your cheek. Okay, it's like you just have to just tickle it out. It's nothing like scrap it out of just dug inside your cheeks. No. Okay, so you are fine, Angie. Hello, Rutangi. How are you? Okay, great, good. No, Avikalp did it at 2. So Avikalp, you are new to my class or you have changed your name? You did it for same mitochondrion. Okay, Chinmay, which stain do you use? You're new, right? That's what I want to ask. So welcome to this foundation class that we have here. Oh, so better not to use that toothpick for your tooth. No, not at all. For mitochondria, you have to use safranin. No. Chinmay, you had did it. Not methylene blue. Is it methylene blue? Iodine. I'm asking for we put eosin. Okay, but I'm not happy for anyone answer. For seeing in mitochondria, you use methylene blue, you use eosin. It wasn't clear. I don't think that stain should be used for seeing mitochondria because you can't see mitochondria using eosin either or iodine either or whatever methylene blue. Animal cells, I'm talking about mitochondria. Mitochondria cannot be stained very easily or with any dye. There is a very specific dye used to see mitochondria, and I give you a hint that is green in color. If that okay, that's why I'm just asking for Chinmay. He did that. That's why I'm asking. But the but the answer for all, you have only one. You want it for one, in 1 month. Anita, not acetocarmine. This is, the important stain that actually used to see mitochondria, genus green. Okay, genus green. So remember it, fine. And Chinmay, ask your teacher to use genus green if they have. Okay, I hope so they have, because you cannot see mitochondria by staining them like this. It has to be, yeah, it has to be, the mitochondria has to oxidize. So, this genus green is being oxidized. Then after oxidization, it gives green color. Okay, that's how you can see green color mitochondria in a cell. Fine. Yeah, no, for cheek cell, normally you are staining your cell outside the cell. Fine. So for that, methylene blue is fine. Okay. But to see inside mitochondria that is very, very smaller than your cell, you has to use genus green. Okay. That is actually oxidizing the mitochondria. Fine. All right. Okay. Fine. This was some extra information. Let's just start with the cell cycle and cell division. Right. So any problem before that? No? Should we start with it? Okay. Okay, so I got today, how many attendees are there? I got 15 attendees here. Anyone who is missing here? I think I'm missing 2, 3 here. Is it? Exactly. Anand. How can I miss him? Yeah, exactly. Michelle and Anand. It can be. Rebecca is here. Yeah. Ashutosh. Okay, fine. So I think they'll join me. Can you tell me what is cell cycle and cell division? Yes. No one is telling me, or some are actually writing. So cell cycle is activities performed by cell. So you understand what we call our normal life cycle. Okay. We say this life cycle. Okay, in which we give birth. We are born actually. Look, Ashutosh is here. Okay, yeah, right, great. Then we become, younger. Then we become mature. Then we'll divide. Okay, then we'll divide, and then one day we'll die. Right? That is our life cycle. That's that's what is being going on in the cell also. Fine, because cell is the fundamental unit of our. You understand, right? It's the fundamental unit that we have inside our body. Fine. It is also having a life cycle. Okay, it is also having a life cycle. So if we talk about life cycle, yes, Chinmay, I understand. There's a series of events that takes place in a cell leading to its division. Okay, it's cell cycle. So normally, for me, a cell cycle is like, having born. Okay, so you get, you're born, then you grew up, then you divide, and after division, it's that you have 2 cells. After division, you have 2 cells. Okay, then you die also. That is an overall cell cycle that what I believe. But in normal books and this thing, the cell cycle is related with cell division, in which only the division, this part of cell is being taken. Okay, when a cell divides into 2 and 2, 2 or 4 daughter cells. That is called cell cycle and cell division. Cells can never have sexual reproduction. Ashutosh, who said this? We have to do plastics today. I know, but we'll take this plastic some other time. Okay. First of all, let us complete cell cycle and cell division. Right, Pranav? Do you have any problem? Can we do it some other day? There is only one topic, plastid, and you have started it actually. No? Yeah, exactly. Do tell me to do plastid in cell doubt session. Okay, Pranav, it's your job to tell me. Fine. Be there on time and tell me, sir, we have to do plastid also. Fine. All right. Yes, thank you. So, what I was talking about, cell is being divided into 2 or 4 daughter cells. That is called a cell division. Okay. Now, what are this chapter? No, this chapter is not at all boring. Who said that this chapter is boring? Until and unless I'm telling you, I'm not finished it. How can you say it is boring, Prajwal? Let me just complete it. Then you will tell me it is boring or not. Fine. Okay, what I was talking about, then because it's the heart of biology, cell division, it's the heart of biology. How you grow up, how you divide, how your cells are being divided. Okay, so it's the heart of biology. How you can say that it's boring? No, it's not. Okay, IKR means I know, right? Is it? I want to improve my abbreviation vocabulary. Okay, great, super. Thank you. Yeah, I know, right? Many drawings are there. That's what make it easy to understand, Rotangi. Okay, Anita, that's good. So let's move on to the first topic. What is cell cycle? So the division of a single cell into 2 daughter cell, or 2 to 4, if we can say 2 to 4 daughter cell, can be said as cell cycle. Fine. So can be said as cell cycle, right? Yeah, they can be many things. Okay, that is reproduction. But you are saying binary and multiple, Prajwal, that is reproduction. Do understand the division, do understand the differences between them. Cell cycle is division of cell, okay, and for normal unicellular organism, cell cycle is only their reproduction, right? So that's what is being, we have been taught for many, I think for many decades. Why only daughter cells? Why not son? That's a good question, and that is always being asked by me. But today I don't want to answer it. I'll give you, is that homework. You find it out, and I'll check it out. What are your answers? Doctor, are better. Okay, I'm a son. I'm not better. Is it that? There is a specific reason. Okay, there is a specific reason they are called daughter cell. I am a man. Why I can't be a son to my parents? So 2n chromosome divided to n chromosome. This is cell cycle. Yeah, this is cell cycle also. Okay. Okay, not to go into this discussion, I have only 1 hour to complete this topic. It's very important. Fine. So, why they are called daughter cells? This will be your homework. Okay. Daughters are female and they only reproduce. Okay. Men, actually, son don't reproduce. Pranav, you are disturbing the balance of earth. Okay. So here it is, cell cycle. But no, before moving into here, we just see what is cell cycle means. So basically, cell cycle are of 2 types, right? They give birth. Okay, right. That can be a reason, Pranav. You're absolutely right. But I want you to, just find it out and then come to me in the next class. Okay, let it be here. Okay, a lot of English, you know, yeah, oh God, seriously. Please, let it, time. We'll take time, okay, fine. In the next class, we'll discuss it in the next class. Someone had posted from Chinmay, don't use Google in here, please. Fahad is trying to log in and log out. Hi Zainab. Okay, so mitosis are of two types. Let's say cell cycle are of two types. That is mitosis and meiosis. Okay, today please don't disturb me. I'm a bit here and there. Okay, fine. Let me teach first. Fine. I was teaching from since morning, so that's that's a problem. Sometimes happen. Sitting here and teaching people and then seeing camera. Okay, that sometimes become very hard. But help me, guys, please. Okay. So what I was talking about, mitosis and meiosis. Okay, fine. It's a very important topic. If you will help me into this, you're going to learn a lot of things from here. Okay, Gaurav sir's birthday. Okay, Gaurav sir was actually not here in the town. He went to his home. That's why we could not able to celebrate his birthday. But he's back, so we'll celebrate. Okay, that's what I'm saying. Don't divert the topic, please. Anita, at the end of the class. At the end of the class, please. Not here. Don't divert me. I got easily diverted. Please. Okay. Let's just a second. Something is gone into my eyes. No problem. No problem. Okay, fine. Cell cycle is divided into two things, mitosis and meiosis. But before going into cell cycle, we first see diploid and haploid organs. No problem. You should come fast next time. DNA is haploid. Do you know the difference between diploid and haploid? Quickly. Anyone? Spelling? I have never heard it. You know about this? 2N and N? Yeah, I know. They are being different in spelling. Okay, fine. I got to know. So let's start from the very basics. Fine. You have cell. In cell you have nucleus. In nucleus you have chromosomes. Am I right? Okay, fine. I understand. Till here it is clear. You have chromosomes, right? Two sets. I understand, Ronak. You are fine. It's all right. Chromosomes. Now, how many chromosomes do we have in our body, or in a single cell? Is it 23 or 46? Is it 23 or 46? Exactly. This is 23 pairs. That is 46. If we are saying pairs, that means 46. Why we are calling it pair? Why we are calling it pair? It's because of the reason they are being in pair. It's because of the reason they are being in pair. One is from mummy. One is from our mummy. Okay. And another one is from papa. Okay. And another one is from papa. Okay. Yeah. Amma, appa. So one is from appa and one is from amma. That's why. Okay. That's why we have two sets of each chromosome. Right. That's why we have two sets of each chromosome. That's why. So basically we should have 23 chromosomes. Okay. we have 26 chromosomes, oh sorry, 23 chromosomes, but we have a pair from each of them. That's why we make 46 chromosomes. That means in each cell, exactly, in each cell there is equal amount of chromosome, equal amount of DNA is present. So that means there should be equal chromosomes also present, because DNA actually makes your chromosomes, right? Okay. So what I'm saying here, that you have two sets, fine. One set is from your father and one set is from your mother. Okay, oh sorry, yeah, your one set is from your papa and one set is from your mama. Okay, fine. So exactly, so what I was talking like this, let's say one, you have a pair of shoes, right? You have a pair of shoes. So basically, let's say one shoes is being bought by your papa, and another one is being bought by your mama. Okay, so what is happening? It is father, it is mother. So let's say you have 23 pairs of these shoes. Okay, let's say you have 23 these shoes. Okay, so half is from your father, half is from your mother. Okay, that is what is being going inside your cell. Fine. So here it is. One set, any one set from your father or from your mother is known as haploid. One whole 23 chromosomes, maybe your father, maybe your mother, we don't care. Okay, it is haploid. It is known as haploid. That means you have half number of cells inside your body. That is what gametes are. So if we talk about gametes, that means either sperm or ovum, they should have 23 chromosomes only. Am I right? They should have 23 chromosomes only. Right? That is called a haploid, a haploid set of chromosomes. Right? And when these two, I don't have, okay, fine. When this two sets meet, one set from, your papa and one set from your mama, okay, it will become 2n. That is your diploid. So di means 2. So 2 haploid, yeah, you can say 2 haploid is your diploid. Okay. Each gamete will have 23 chromosomes. Exactly, Anita. Absolutely fine. So now you understand, in your body, half a haploid set is from your mother and a haploid set is from your father. Right? Understand? Any confusion in here? Any confusion? Nope. So basically, what we are trying to do in here, mitosis give you diploid cells, or can give you haploid also if the starting one is haploid. Meiosis give you it is always giving you haploid, one set. That means in mitosis, if we are starting with 2n, we are getting 2n only. The daughter cells are also 2n. Okay. Chinmay, the name is like that. I cannot do it. So maybe I don't know. Something I also don't know. Okay. The Buddha people have someday they have named them. Okay. So I cannot say because I haven't named them. I only studied like this. Only. Fine. Understand? What I was talking about? Di and hap. Okay. Yeah, those people in 50s and 40s, when they research about it, they discovered it, they named it. So now a time they become Buddha only. Fine, that's why I call them. Okay. Old people, man, old people, the old chaps. Don't you understand what happened? Okay. Okay. Yeah, we are talking about mitosis and meiosis. Uh, Here it is. If we are starting with 2n here in meiosis, we are getting only n. Fine, we are getting only n, that is haploid. So basically a meiosis is used in gamete formation. So what about the other n? Angie, there are 4 daughter cells in meiosis and there are only 2 daughter cells in mitosis. What if we start with n in meiosis? The meiosis will not take place. Mohammed, the meiosis will not take place. Fine. Okay. Alright. That is the difference between mitosis and meiosis. We'll go and see it. But the cell cycle is overall divided into interphase and M phase. That means a mitosis, I'll take a pen, a mitosis or meiosis. You understand? So both have M in the starting, that is why we call them M phase. Alright? Is it fine? Now, this interphase is divided. If we start with N mitosis, then what will happen? Rutangi, I just answered it. Where were you? Who is going to answer this? Who is going to answer this? Oh, what do you ask? Oh, you asked mitosis. I'm very sorry, Rutangi. I'm really sorry. I think you asked meiosis. Yeah, if we start with, if we have N in mitosis, okay, all the daughter cells will also be N only. Sorry, it's only 2. Fine, all the daughter cells will only be N. Okay, sorry Rutangi, I thought you asked meiosis. Okay, interphase, the G1. Okay, the G1 is the very first phase in the, so let me tell you. How will I tell you? Okay, so this is like this. So it's a cycle. It's a cycle, right? This is your first interphase. Okay, and then your M phase. Yeah, the interphase that it suggests inter means in between, right? In between. So interphase is nothing but a phase, a resting phase of your cell in which it prepares itself for the next division, right? Understand? So it's a preparation phase. So it's a preparation phase, the interphase, right? And then when all things are prepared, they'll go into M phase to divide, right? Now, what are the preparations here? So if you go and if you do marriage, you have to prepare also. Fine. For doing marriage, you have to arrange all the things, all the caterings and barat and all sort of things. So that is a preparation. So what is the need for this preparation? Why do we need to prepare? Okay. First of all, I told you if you have 2n, let's see the case of only mitosis. We'll not go into meiosis. Meiosis is a bit difficult. Okay, mitosis is very easy. So for mitosis, also 2n and 2n will be forming. But here we are only having 2n. Where this 2n is coming from? Tell. Say guys, where this 2n is coming from? Okay, let's say I have 10 rupees in my pocket. Okay, and Anita and Kashif come to me, sir, give me 10 rupees. Fine. I want to have an ice cream or chocolate, whatever. Fine. How will I can give them 10 rupees if I have only 10 rupees in my pocket? You understand the question? I can only give 5 rupees. That is what, you know. Please don't make me beggar. Okay. No, that is what I'm asking you. It has only 10 rupees. It has only 2n. Okay. And it is giving both the tortoises 2n, 2n. Okay. How it will give? 5 rupees, 5. But no, he is giving 10, 10. He has 10 rupees and he is giving 10 and 10 here. No, it is 2 and 2n only. Don't think about my money. Think about the chromosomes. Think about the thing which I talked, just taught you before. Zainab, don't become, ACP Pradyuman. You should have said, kuch to gadbad hai, Daya. Rebecca? Where is Rebecca? Run away from the preparations. Rebecca is not here? Okay. Rebecca, anything you want to add? Exactly. Avikalp. That is what I was asking you. That is what I want asking you. DNA has the property to double itself. Guys, replication. DNA has a property to double itself. It can make its copy. It can make its clone. Then what's the problem? It's not about my 10 rupees. That's what all of you are thinking about my 10 rupees. How will I give you 5 rupees? That's what in your mind, but you are not thinking about the DNA. DNA has the power of replication. DNA has the power of cloning. DNA has the power of doubling its DNA. Yeah, you Avikalp. Okay, great. So the replication of Zainab, thank you. Replication of DNA starts. So basically, if you want to divide, you need to give equal thing to your daughters, right? Okay. Otherwise, your daughter will fight. So that is what is happening here. They have to give equal DNA to each one of them. Okay. And for that, you have to divide your DNA. You have to divide your chromosomes. So if you have 46 chromosomes, so before division, it will become double and become 92. Okay, fine. Yeah, Shutosh. So you people are very always. You people should take commerce after your 10th class, because you people monetary, not thinking about my cell, not thinking about my my little cell here. Okay, fine. It's like for 10 rupees you people are fighting. What if I have told you, I have 1,000 rupees note in my pocket and I have to give you, and you'll just burn me out. Okay. What I was talking. Okay, replication of DNA is one thing. Fine. And and then what I'm asking? Alright. Then there is also division of organelles, like your mitochondria. Okay, fine. Like your mitochondria. Exactly. Fine. They also has to divide. All right. And then the cytoplasm has to enlarge so that it will go to each and every daughter cell. Yeah, centrosome also. Okay, Golgi, everything has to get doubled. Okay, So that these things are being these things are being actually doing in preparation period. So there is G1 phase, okay, there is this G1 phase, there is S phase, and there is G2 phase. Fine. So after M phase, first there is G1 phase. So the first phase is called G1 phase, then there is S phase, then there is G2 phase, right? Okay. Now M phase. So in M phase there are two things. Karyokinesis. Okay. Karyokinesis means division of nucleus. Okay. And cytokinesis means division of cytoplasm. Right. G1 is after the division. Okay. G1 because it's the first gap phase. G means gap. Okay. So it's the first gap. So after with division, the very first gap phase, the very first phase is called gap 1 phase. Then there is a synthesis phase, S for synthesis. Then there is gap 2 phase. Alright. Now karyokinesis is also divided into 4 phases. That is your prophase, that is the first phase. Then metaphase, that is the middle phase. Anaphase, next to the end one. And the telophase, that is the tail or the end one. Fine. You understand how they are being named actually. Is it, guys? Prophase. I'll go into define each and every phase. Not a problem. Why you people are only worried about prophase? Why not what happens in telophase and anaphase? Let me teach first. Okay, so here it is. Fine. So this is what is being taught here. So this is G1 phase, this is S phase, and this is G2 phase. Fine. And this is your M phase that is working here. Fine. So let's see what are happening in different phases. These are the phases of growth in which your, normally G1 is big. Okay. And it is it starts to prepare intermediate. Your mitotic phase is very small phase if you compare with your interphase. Fine. Because in mitosis, have you seen mitosis in have you what's there, have you seen mitosis in microscope or any other cell division in microscope? No. Okay. Okay, great. So if you have seen, there is a lot of stage. Okay. So why there is a lot of stage? Because the single stage will only last for some seconds. Okay. The single stage will only last for some second. So that some cell will be in prophase, some other cell will be in anaphase, some other cell will be in telophase. So it only lasts for some seconds. Okay, fine. People, please don't discuss among yourself. That distracts me. Understand? Please don't talk among yourself. First, let me teach you. Okay, fine. What I was talking? Yeah, phases. So basically your M phase is small, then your interphase. So in G phase, S phase and G2 phase, cell prepares for the cell division. So let's see what happens in which phase. So interphase, period of intense growth and synthesis. So growth of everything like cytoplasm, like proteins, everything is grown inside everything is synthesized in your cell. Fine. Most active stage of cell cycle. It is most active state and takes 75 to 90% of entire generation. Fine. That's why interphase is the longest phase of cell division. Okay. Interphase has G1 phase, S phase and G2 phase. So let's see what are these phases. G1 phase, growth of cell takes place. Now think that two cell, a single cell is being divided into two cells. Now this cell will also divide. Now think about this cell. Cell number 1. Okay, this cell will also be divided. Now this cell is usually smaller because it has just divided. Like your baby, it is smaller. It has to grow first. So it will grow. Okay, that growing phase is your G1 phase, right? This phase occurs between the end of mitosis and initiation of DNA replication, right? Now the next phase is synthesis phase. That is why it is called synthesis phase, because DNA is being synthesized in this phase. DNA is being synthesized in this phase. But you need to understand, DNA will not synthesize automatically. It needs to have many proteins and enzymes. It needs many things. For that thing, the G1 phase actually prepares for synthesis phase. Right? G1 phase prepares for synthesis phase, because a very important thing in cell cycle is the division of DNA, is the replication of DNA, is the doubling of chromosomes. So in G1 phase, it actually prepares cell, okay, and it grows the cell also. Fine. For accommodation of double nucleus. You understand? They have not double nucleus but double chromosome, double DNA. So a cell should be bigger. You understand? A cell should be bigger to attain that thing. Okay. Fine. Okay, just a second. Something happened. Okay, fine. So this is S phase. So this stage of replication and synthesis, the DNA content doubles but the chromosome number remains the same. This is the question. The chromosome number remains the same. So what is happening? What is happening that you have this? Yeah, that's what I'm telling you here. Okay, this is your chromosome. Okay, let me tell you. How many hand do I have? 2. How you can say 2? How you can say 2? How many hand can you see actually? You can see only 1. Yeah, but there is 2. Exactly, there is 2, but at a time you can see only 1. At a time you can see only 1. Okay, fine. Aditi, what you didn't understand? So let me tell you one thing. So basically this chromosome doubles, but you can see only one type because the other one is behind this. You understand? The other one behind this. DNA content double, Arunav. DNA has to double itself. So if you have, if you have a DNA for 46 chromosomes, okay, so you have to double itself now, so that you have a DNA for 92 chromosomes. Fine, understand? Okay. Then we have G2 phase, in the preparation for cell division takes place. Okay, and synthesis of protein required for mitosis occurs in this phase. That is what being going on in interphases. Now next, but here 46 to 92 number is changing. We have 46 chromosomes, and if you double it, that is a simple maths. That is a simple maths. Chinmay, any problem? Fine. This follows the interphase. It aims at orderly distribution of already duplicated chromosomes between two daughter nuclei. So in mitotic division, now the division will going on. Second point of S phase, the DNA contents double but the chromosome number remains the same. That's what I told you here. The chromosome number will remain the same because they remain intact with each other. So all you can see is 46 chromosomes only. Chinmay, you didn't understand? All you can see is 46 chromosome at a time. Yeah, because in one there is 4. Here you can see 22 only, but there are 4 actually. So the chromosomes are actually, you can see they are actually, how 46 into 2? You didn't got it. Okay, let me tell you once more. So basically, what I told you, I have 46 chromosome in a cell. Okay. In daughter cells, there should be also 46 chromosome. That also disturbs. Okay, that also need 46 chromosome. How can it will give you 46 chromosome without doubling it? So in between there is a phase in which it has to double itself so that it becomes 92. Okay, and then give 46 to each one of them. Okay, right? Yeah, double means it is multiplying, it's cloning itself. It is cloning itself. So same chromosome, okay, is cloning itself and giving its clone just behind. Like this is my same hand. It is cloning hand another and it is just behind it. So you can see overlapped. Yeah, exactly. Now understood? That should hate cloning. Okay. Now there's one more phase that is called G0 phase. In this phase, it happens when a cell not wants to divide. So it goes in G0 phase. Fine. It is called the quiescent stage. That means the resting stage. Okay. The cells remain metabolically active but no longer proliferate unless it is called on to do so. What Pranav? What interface? So basically, let's say it's M phase, it is interphase. So when a cell come to interphase, it goes to another that is phase, that is G0. Okay, proliferate means it doesn't proliferate, doesn't grow in size. Okay, it doesn't grow in size. That's what I'm saying. It doesn't grow its size. It's remain metabolically active means all the function it is performing. It making protein. It is doing all the task. It may be a let's say it's a cardiac cell. Let's suppose it is a cardiac cell. Let's suppose it is a neuron cell. Fine. Let's suppose it is a neuron cell. So it divided. Then it is doing its normal function. Okay. It can do its normal. Anita, just wait a second. It's normal function. Don't write it down, Anita. You can see it in recordings also. Fine. So what I was talking about, neuron, right? So neuron does normal function. Okay, so it's remain in the G0 phase, but it wants to divide. When it wants to divide, it comes from G0 phase to this phase. So in this phase, in G0 phase, it does all the functions except it is not proliferating. It is not growing in size because it doesn't want to divide. Understand? Yeah, there is. Okay, Rita, this was the last slide that you want to ask me. So I have just did it for the last time. Okay, fine. Quiescent is your resting stage. Fine. All right. All of you quickly tell me so that I can move on. There is one raised hand. Aditi has raised hand. Okay. Which cell do not undergo cell division? Back, permanent tissues. No, not RBC, Angie. Dead cells. Okay. So I'll ask which living cell do not undergo cell division or undergo cell division. Viruses also can divide. Neurons. Yes. You know, if it starts to divide, your brain will become like this. Okay, each and every year your brain will become, you'll become like Mojo Jojo. Have you seen it? I don't think so. You have seen that. Have you seen Mojo Jojo? Yeah, exactly. Powerpuff Girls. Yeah, so that's what, you know. It's also, when I was also a kid, I used to, you know, see Powerpuff Girls. So in that, I was having Mojo Jojo, and there was one more character, Amoeba Boys. Have you seen Amoeba Boys? So they are like amoeba, okay, and they always like, there is always cyclosis motion is being, they move like that. Yeah, I know. SWAT cats. Yeah, exactly. I have seen in my childhood also. Actually, I was also small someday. Yeah, yeah, your topic I know. Mitosis, coined by Wather Fleming in 1882, common method of cell division in body cells and mitotic cells. So, what is this? Mitosis so famous for? Got cut. You got, you want to grow in size, okay, and you become fat also. These all are actually done by mitosis. So you got cut in your hand, okay, and then what happened after some day? It just goes well, fine. So what is it being happening? What is it going on? It's mitosis. It's the division of your cell that makes you healthy, that makes you normal. Fine. So that is your division. Somatic is your normal body cells. All the cells except your germ cells, except your sex cells, are called as somatic cells. Fine. Your body cells. Okay. And it is called as equational division because 2n will giving you 2n again. The equation is balanced. Prajwal, take your time, enjoy this thin period of yours, okay, because when you grow old, you are going to just remember those nostalgic moments when I was so much thin, because I also used to imagine my days of thin when I used to have 28 waistline and many more things. Just enjoy this part, please. I don't anything. Yeah, right. So exactly. Okay. Next. So M phase, there is karyokinosis and cytokinosis. So let's start with very first phase in the anaphase, telophase, prophase. So let's start with it. A platelet's the cell that undergoes mitosis. Arunav, normally your cells that you are in your body, they reproduce or they divide through mitosis only. Fine. Platelets also. Cellulite actually dividing cells? What is cellulite, Chinmay? I didn't get it. What is cellulite? Fat. That's what I want to add. Does fats are cells, or you are asking about adipose? So that's what I want to ask. Fat cell, yeah. We are talking about adipose. So fat cells, why we are calling them fat? Because in there is a storage of fat. There is not division. So whenever there is a storage of fat, that's what make it fluffy. That's what make it bulky. Okay, fine. In there any part after during mitosis involving membrane biogenesis? There's a lot of part Avikal. Okay, and the very first thing is your nucleus that needs it. Okay, we'll get to see it. Fine, we'll get to see it. Okay. So let's see. The first stage of mitosis, that is your prophase, following interphase, usually the longest and most complex phase of cell cycle, in which it is not, it doesn't look like this. Your chromosome doesn't look like this. It is more like a thread-like structure. Okay, okay. And in this phase, your DNA start to condense and form chromosomes. Okay, your DNA start to condense, your chromatin. Exactly, chromatin entangles, chromatin condenses and form chromosomes. And I can you know, you can easily understand this thing. All those girls, if they have a hair, if they have a long hair, okay, you do brush your hair, you get a lot of what I'll say, hairs, okay, and you just entangle it, so it make a, you know, a ball like, you know, what I'll say, a ball like hair you can see. Am I right? You know, whenever you you know, do brushing, right? So you have a long hair, and then what you do, you just, you know, condense it, twist it, you just coil it, okay, and it becomes a ball like, you know, it become a, you know, a thick structure. That is what is being going on inside the nucleus. The DNAs are long, okay, it start to condense, okay, it start to condense and become a ball like, a thick like structure. That is your chromosome, right? Understand what is being going inside the, you know, inside the nucleus. Yeah, what I told in the last class, that hair thing, I don't think so. This is what I got right now. What no? Oh, visible while dividing. About DNA. Okay, fine, all right. So usually the longest and most, so in this phase, what happens? Your DNA start to start to condense and start to forming chromosomes, and then your centriole divides. So you can see there are two centrioles. So in normal cells there is only one centriole, okay, but and this is also present in only animal cells. Okay, centriole is also present in only animal cells. So that is also divided and goes to two different poles. That defines a pole. Yeah, exactly. Not in plant cell. All right. And your nuclear envelope here, your nuclear envelope, you know, start to thin off, start to, you know, just vanish off. Fine. Animal pole and vegetative poles. Acha. Okay. Fine. Centriole is not present in plant cell, Rutangi, like chloroplast is not present in animal cells. Like the same way, centriole is not present in plant cell, right? Aviculp is you know, it forms spindle. Sorry, what I'm talking about, spindle. It's formed astral rays. Fine, your centriole, it forms astral rays. That's why we called animal cells as amphiastral. If you people remember. That's why we call them amphiastral, and plant cells are called anastral. Okay. Yeah, there is division of plant cells. Who said there is no division of plant cell? Do you think plant, you know, cells only divide through centrioles only? The division is because of you know, dividing of chromosomes, not division of centriole. Understand, Aruna? So next we are moving on. Prophase. So prophase is also divided into you know, different phases. That is what, Fahad? Fahad said something. Same, Anita. Oh, I think you people are conversation among so, oh sorry, early prophase, mid prophase and late prophase. Fine. So these are the different stages. So you understand that when a cell divide, it doesn't have a stoppage, a stage. You know, when the prophase will end, okay, then after 2 hours it will start. It will start next phase, that is your metaphase. Then when metaphase will end, then after few moments it will start anaphase. Right? That will not happen. It will goes like in a same you know, in a same process. So you cannot able to find the difference between what is, you know, when is prophase is starting and when phase is metaphase is starting. How can plant cell divide? They have cell wall. It's very rigid. How can they divide? How can plant cells? Okay, people are actually just copy pasting your question. Plant cells also divide. So first of all, it doesn't matter they have cell wall or something. The thing is going inside the cell. Okay, the thing is going on inside the cell. Their cytoplasm divide. So if this is a plant cell, okay, there is cytoplasm divide, okay, and in between this, a new cell wall is also formed. Fine. Okay, no problem. Fine. So let's move on. So early prophase cell becomes round and cytoplasm term more viscous, means more thicker. Okay. ER and Golgi apparatus break down. Astral rays in animal cells are formed. Initiation of assembly of spindle microtubules. Fine. Now spindle microtubules, they are started to form. Okay. They are started to form inside the cell. Viscous means thick. Viscous means thick. How I will, have you seen water and honey? Water and honey. Water is less viscous because it can flow easily. Honey is more viscous because it is more thick, so less more penetrable. Water. Fine. All right. Viscosity, exactly. Now you can easily understand what is viscous. What are astral rays? The rays which coming from your centriole. Okay, they are also proteins. They are also microtubules. We're going to understand it later. Chromosomes attain characteristic size and shape. Okay, nucleolus starts disappearing. I cannot go back. Please, Pranav. I have very less amount of. I have only 15 minutes left. Late prophase, spindle formation is initiated, and centriole pairs and push to opposite spindle poles. Sorry for that, but seriously, Pranav, don't mind, but I don't have time left. Now this is metaphase, and metaphase is the best phase to see chromosomes. First of all, they are highly condensed. Okay, they attain their characteristic shape, and then they are also there at equator, at the center of the cell. Fine, they are at the equator. So spindle moves into the middle of the cell. So you can see there the spindle are coming out from here. Okay, and they move into and they bind to one from here and from here. Okay, they are bind to one chromosomes. Right, chromosomes get arranged along the equatorial plate through spindle fibers. Arms of chromatid trail away from the kinetochores. Now, what is happening is here, this is. Let's say it is a chromosome. Okay, let's say it is a chromosome. The spindle fibers are now and come bind to it. Okay, and then it will push this chromosome towards itself. Fine. Kinetochore. This is your centromere, and there is a protein here and here. Okay, let me. There is a protein here and here. Okay, with each chromatids. Okay, with each chromatid there is two kinetochore. This helps to bind with spindle so that it will take both the chromosomes to the opposite end. Fine. Next, what is the best stage for studying mitosis? Ashutosh, that was centriole. Exactly, metaphase. So I told you metaphase is the best stage for studying, I think not mitosis, but here it should be chromosomes. Fine. Because mitosis is the whole cycle is mitosis. Fine. So I think the best stage for studying chromosome is metaphase. Right. Anaphase. I corrected Pranav. You see. Fine. So sister chromatids separate at kinetochore. So you have, then you will ask me, sir, what is sister chromatid? So they are sister to each other. same chromatids, they are called sister chromatids. So this is one sister, this is two sister, and please do not ask me why sister, why not brother. I'll give you this at homework, if you're going to ask me. I don't know why they name this. I do not. They must be, I don't know. Chromosomes. Yeah, Rakhi in the center. Exactly. Thank you. Chromosome are pulled VLJI shape. Okay, fine. As chromatids move apart toward their respective poles, the spindle fibers elongate and cells become larger. So that's what is going on here in anaphase. You can see here. Okay, the chromosomes which are being present here in the metaphase, okay, they are being pulled by these spindle fibers at the poles. Now they are being pulled by the spindle fibers at the pole. Understood? The next, what causes the movement of chromatids? Yeah, spindle fibers pulling. Exactly. Spindle fibers pulling. What is the advantage of condensation of chromatin during cell division? Now, what is the advantage? Easy to divide. Now, the scenario that you want to relocate your house. You have taken a new house. Okay, you have taken a new house and you have to relocate. Fine. What you will do? You will pack your bags first in your bags, right? So that it is easy to transport. You will not, like your clothes are there in your, shoulders and you're going like that only. Your, almirah is in your hand. No, you will not do that. Fine. So you have to condense everything, right? You have to condense everything. You have to pack everything. So that packaging of DNA is your chromosome. That packed DNA of your cell is your chromosomes. That's why formation of chromosome is so much important. Now telophase, a very easy phase is that it's just opposite to prophase. Okay, it is opposite to prophase. So all the cells go to all the chromosomes go to different poles. The nuclear envelope the nuclear envelope again what I'll say. The nuclear envelope again form. Fine, this you can see here. Okay, nuclear envelopes again form. Okay, and all the things are restored back. Chromosomes uncoil and become long and slender. Nuclei start synthesizing ribosomes. Spindle fibers and astral rays disappear and cell resumes original shape and size. Okay, fine. So it is, I said it is just opposing to the prophase. Yeah, that's why I make it, guys. That's why I make it. And don't tell me that it looks like my face. Otherwise, I'll be very angry. Okay, then what is the difference in animal cell and now this division after this nucleus division, the division of cytoplasm takes place. So it is like from outside to inside. Okay, so there is the cytoplasm are divided like this, but in plant they are from inside to outside. Okay, that's what is being which you are seeing here. Okay, this is the cell plate is being formed. Okay, a cell plate is being formed, that is also known as phragmoplast. Your cell plate. This is the plant cell. Okay, fine. Which outer covering? Which outer covering? Cell wall. Now, significance of mitosis. Maintenance of cell size, growth, genetic stability of by maintaining chromosome number. Okay, so we all have 46 chromosome in our body, so it maintains that number all through our body cells. Okay, replacing old cells with new ones. Okay, like your RBC has only 90 days 180 days 180 days life period. Fine. So after 180 days, after 120 days, what happened? After 120 days, what happened? It will die. So it has to be replaced. That replacement is also by mitosis only. Healing and regeneration. So you got cut, you got burned, you got everything. It got healed. It got regenerated. Yeah, 4 months. Reproduction. Okay. So reproduction is also very, your zygote, it divides. Okay. Your reproductive system is also worked by mitosis. Apart from making of germ cells, apart from making of sperm and ovum. Fine. Establishing evidence of relationship. Okay, that I don't know what is this. Sorry. Establishing evidence of relationship. Seriously, I don't know what is this. Why it is written here. Okay, so I don't know. Seriously. Rest of anything, if you didn't understand, you can ask me. Okay, fine. Yeah, it sounds social. So cell division is clear to you. I think when you study, meiosis also, that will going to clear a lot of things. But meiosis is not in your syllabus. In 11th class, you are going to study. That's not a problem. You don't know, Ronak, who made this slide. How I can tell you? Yeah, Aadhaar of relationship. Okay, yeah, it is finished. It is finished. So what factors induce mitosis? You don't have to know. Yeah, Anand is pretty upset with me. So any doubts? Why, Ashutosh? Chinmay will do meiosis in in 11th class. No, it is not hard. Yeah, KKR lost. Good. First time seeing thank you. I like PMC. Then take it, no problem. Thank you all of you. Okay, so I have less time. So any doubt, please, apart from IPL. Thank you, Rebecca. Ashutosh, what RBC? No doubt, nothing to ask. Then we'll be meeting next time with a new topic. Yes. Red blood cell. What red blood cell, Ronak? Tell me, what is it? Gaurav's birthday. Yeah, we'll celebrate it. RBC. Yes. I'm not seeing IPL. I don't have time to see IPL actually. Okay, I'll tell him. Okay then. You don't have anything to say to me, then I'm going. I'm really hungry. Please. Okay, fine. Bye bye. Take care. I'll tell you in the next class. Bye. See you.